Right, cool. Now we are live. Guess what, everyone? I thought we were live 2 seconds ago and we weren't. But now we are. And I'm really, really happy about that. May I introduce David Henshall, who is our guest of the week and last week on the Epilepsy Sparks Insights podcast. And David is you basically don't miss the episode because the episode is very interesting about microRNA and how our understanding has come on over the past 30 odd years, I think. But David, who are you? Tell us just a little bit about your background, please.
Yeah, so hi Tori, thanks for having me on the show, and thanks for the interview around the book and the work that we've been doing. So I'm a professor in the physiology department at the Royal College of Surgeons in Ireland, or RCSI, University of Medicine and Health Sciences. We're a big medical school in the city centre here in Dublin in Ireland. And my research concerns epilepsy, and in particular I've always been interested in trying to understand how activity of genes within the brain changes in epilepsy, and to try to use that knowledge, that understanding, to design and develop drugs for the future.
And your book, what's it called again?
The book is called Fine-Tuning Life, with a subtitle of A Guide to MicroRNAs, Your Genome's Master Regulators.
Okay, now that sounds rather wash and kind of suitable for a particular audience. Could you break that down for us? So what does that mean in layman's terms? Although I will say you don't have to be a scientist to read it, because I've started reading it, and even my little brain can get round it. So.
Yeah, so the book is really about this very special and unique type of gene, which was only discovered a relatively short period of time ago. We have been working for many, many years and understanding a particular type of gene we called a protein coding gene. So these are the traditional genes that we all talk about. But hidden within our genomes are a whole other type of gene that's absolutely essential for how cells work, and ultimately how organisms, including humans, work. And so this is the type of gene that we've been studying for the last decade or more in my lab here in Dublin, as well as others around the world. There are actually a number of groups in the field of epilepsy that have been studying this type of gene and trying to understand its role, its importance, and whether or not it could be a target for therapies in the future.
If somebody tries to think of genes, DNA, we often think of like curly whirly pretty thing with little horizontal sticks in the middle. But when we type in if anybody fancies this, do it. You go into Google pictures and you type in microRNA, and it comes up with what looks like half of a DNA. Could you explain that, please?
Yeah, okay, I'll try and do this quickly, right? So our DNA is this classical double helix. It's sort of two strands really wrapped around one another. And a gene is really the information, the instructions to build proteins. In order to build a protein, our DNA actually has to unwind. It sort of unzips a bit like a zipper. And one side of that, if you think of the teeth of the zipper, is a sequence of 4 different type of chemicals called bases. And really the genetic code is this really long sequence of one of these 4 different bases. But we don't jump straight from DNA to protein. There's a molecule that carries the information, the kind of code in our DNA, to give the instructions to build a protein. And that molecule is ribonucleic acid, or RNA. A particular type of RNA called mRNA. There's actually a few different types of this RNA. mRNA basically carries the information read from the DNA code, and then takes it and uses it to translate into what we call a protein, which is a long assembly of amino acids. So information goes from DNA, gets translated into a code that assembles amino acids into proteins. And microRNA are a different type of RNA, also coded within our genome, but they sort of regulate the amount of the protein that gets produced.
And it's that regulation of that protein which you talk largely about in our episodes of the podcast, because it's essential to keep. You explain how it's essential to keep the levels at a nice sort of state, in a stable sort of, I don't know, what would you call it? Just in the right place, so that things don't go skew-whiff and muck up one's brain activity and cause potentially seizures.
Yeah, so what we know is that gene activity kind of can occur in sort of little bursts and has a tendency to be a little bit overactive. So hundreds of millions of years ago, a separate type of gene kind of evolved alongside it that worked as a regulator, and it sort of dampens down the activity of genes, keeps things stable. It's a sort of a feedback loop. And so microRNAs are really this sort of the genome's solution to noisy gene activity.
Or like the conductor of an orchestra to keep everybody playing the way that they should, correct?
Exactly. This is one of the kind of metaphors for what microRNAs are doing in our genome, so that you can think of DNA sort of as the music on the page, and the kind of the RNA and the proteins are sort of your musicians and instruments. And what a conductor does in an orchestra is tells everybody kind of when to start, when to stop, how loud to play, how quiet to play, how fast to play, how slow to play. And you really need that for a piece of music to work properly. And this is essentially what we need in our genomes. We need a system to sort of quieten things down, make things a little bit louder, speed things up, slow things down. And there are systems that allow genes to do that. And microRNAs are really critical for that process to work properly. And we know it's critical because if we lose microRNAs, if there's a mutation in a microRNA gene, it really upsets gene activity in cells.
And we find, you speak about how you found, like, for instance, in Dravet syndrome, this is the case, correct? Which is a rare epilepsy.
So
Yes?
at the moment, we've been trying to see if some of the genetic epilepsies have mutations in microRNAs that cause them. We haven't got evidence for that yet.
Okay.
There are groups around the world that are looking at the sort of the genetic code of patients with some of the rare epilepsies to see if there are mutations in microRNA genes. That doesn't seem to be the case in general at the moment. What we have done a lot more research on, and where we do see much more evidence, is the more common acquired forms of epilepsy, one of which is temporal lobe epilepsy. So if you analyze, and there are ways to do this, if you analyze brain cells within the part of the brain that triggers seizures in temporal lobe epilepsy, you do see changes in levels of several different microRNAs. Some are a little bit higher, some are a little bit lower in that brain tissue, which would be predicted to alter the activity of the protein coding genes there and cause problems.
And so tell us, why is it cool and exciting to know of the professional journey of learning about microRNAs over the past couple of decades? Why is that? To me, like, the book is very exciting. I get it. But to people who might not be familiar might think, like, who cares? Like, I just want to know how things are right now. Why is it cool to understand what has happened?
So I think there's several things. Like a lot of discoveries in science and medicine, there's a sort of a starting point. There's a period of time where people kind of ignore it. You know, somebody makes a discovery. In this case, the discovery was made in a worm about a particular type of unusual gene, which didn't seem to make a lot of sense at the time. This thing doesn't code for a protein and can't be very important. And things sit there quietly, and then one or two other people make an observation, and then all of a sudden, you know, this discovery explodes, and it sweeps like a wave through the world of genetic research and medicine and biology, and all sorts of people get interested in it, and they then you get this wave of sort of discovery after discovery. And I kind of made this point, and it's made in the interview earlier, that within a short period of time we'd accumulated an understanding of the whole basic process in the space for a few years, and that energy and creativity is a wonderful thing to capture. And then there's this sort of phase afterwards where people begin to take basic discoveries about a system, a new type of gene, and think about ways to turn that into technologies and medicines. And so then there's this second phase where people start to think, okay, well, maybe we can medicinize this and create drugs. And that's what happened. We create, within a few years of discovering that these new genes were in humans, we had a medicine being tested in people. It's amazing stuff.
Yeah, because you look at history prior, that never would have happened. It would take like what felt like forever to even get to that stage. Yeah, it is very exciting. And as I said before, if you've read a few papers, you can read this book. It's actually not layman's terms, but it's written in a way that I think lots and lots of people can understand if you're a reader anyway. And just go for it. The title, remind us of the title again, David?
Fine-tuning life. A guide to microRNAs, your genome's master regulators.
That's right. And if you need more enticing, check out the chat with David and I on the Epilepsy Sparks Insights podcast, which is also on YouTube. It's pretty much everywhere, or on the website, toryrobinson.com. Thank you very much, David.
Thank you, Tori, for having me. Terrific to see you again.
Ditto. I'm just going to stop live.